Hi everyone, this is Maret Lopez Ocasio, and today is March 19, 2025. And then I wrote here my name, Maret Lopez. We're going to be talking about 3 different macromolecules today, and the first one I selected is cortisol. Cortisol is a lipid steroid hormone. This hormone, it starts its synthesis in the smooth ER, and then it gets transported to the final destination of the bloodstream. But step by step it will start as cholesterol. Cholesterol gets modified to produce cortisol. So cholesterol is a precursor of cortisol. Then as a transport step, cortisol gets moved via transfer vesicles to the Golgi apparatus on the cis phase of the Golgi apparatus. And the Golgi apparatus, it gets further modified, like adding functional groups or adding carbohydrates, and then it just gets packaged and transported and secreted through the transphase of the Golgi apparatus into the cell membrane. Then when it gets to the cell membrane, it gets released into the bloodstream via exodiastasis. Cortisol is really important for stress response, metabolism regulation, and even inflammation control. The next one that I'll be talking about is glucagon. This is a protein hormone. Its synthesis, unlike the cortisol, it starts in the ribosomes of the rough endoplasmic reticulum, and the precursor gets made there. Then further folding and modification happens in the rough endoplasmic reticulum. Then it gets transported via transport vesicles to, again, the cis phase of the Golgi apparatus. Further processing, like adding functional groups, etc., happen in this Golgi apparatus, and then it gets secreted via secretion vesicles to the cell membrane as well. Then this secretory vesicles from the transphase transfer that glucagon to the cell membrane, where it also gets released by exodiastasis into the bloodstream. Glucagon is really important in regulating blood sugar, and it transforms glycogen into glucose. One thing that I like to talk about is that it's really related to insulin. So insulin will be the opposite. So when glucagon will cause that high blood sugar when our sugar is low, because glycogen is like big storage of glucose, gets broken down into glucose, and vice versa with insulin. When there is a lot of glucose, insulin helps in transforming that glucose back into glycogen to store that glucose. So they play a big role in regulating blood sugar. The intracellular structure of protein that I selected is microtubules. Their synthesis starts in the cytoplasm. The ribosomes are in the cytoplasm. That's what happens is as these tubulin dimers get formed, and then they rearrange in the microtubule organizing center, also called as MDOC. And then their final destination will be used for intracellular transfer, structure, and cell division. So they stay within the cell. They just form these tubulin dimers, and then they just rearrange in a way that supports intracellular movement. They're really dynamic. They can assemble and disassemble really easily. And again, they get formed in the ribosomes, but the ribosomes in the cytoplasm. Another thing about microtubules is that they are really helpful in transport of organelles within the cell. And again, they're really have a really dynamic nature, and they also participate in a mitotic spindle during cell division. In summary, the movement of macromolecules is essential for cellular function and homeostasis, which is just the balance in our body. Cortisol is a steroid hormone. It's synthesized in the smooth ER and transported via vesicles to regulate metabolism and stress responses. Glucagon, a protein hormone, is produced in the rough endoplasmic reticulum and processed to the Golgi apparatus to regulate blood sugar levels. And microtubules, on the other hand, are structural proteins that are synthesized in the cytoplasm and play a crucial role in intracellular transport and cell division. Understanding how these macromolecules move within the cell really shows the intricate mechanisms that really sustain life. Thank you for your time, and I'm happy to answer any questions.